Hello, everybody. My name is Daniel Suter. I'm a professor in the Department of Biological Sciences at Purdue University. I would like to thank the organizers for giving me the opportunity to talk about some of our work, specifically today about our project on the role of reactive oxygen species in axonal growth and guidance. My lab overall is interested in understanding the basic cellular and molecular mechanisms of nervous system development and regeneration. And in this project specifically, we look at the role of Nox-derived ROS in nervous system development. In two model systems, in Aplysia, Californica, and Zebrafish. This title slide here shows two images. On the left side, you see a cross-section through the eye of a wild-type zebrafish embryo at 2 days of development, two stains, in blue nuclear stain, and in green a stain for retinal ganglion cells, RTCs. You see the nice layering of the retina, and also you can see the nice formation of the optic nerve as the axons come together from the nesting ganglion cells and exiting the eye. On the right side, this looks very different. This is a section through the eye of a corresponding Nox2 mutant. So this fish doesn't have any functional NADPH oxidase 2. This enzyme produces superoxide. So this is this eye is completely different. The layering is not happening, and also the retinal ganglion cells don't form axons together, and they don't exit the eye in the same fashion. So this provides evidence that ROS from this complex are really important in retinal ganglion cell development as well as making connections to the brain. So this is the outline of my talk. I will focus in the background briefly on axonal growth and guidance. I will then cover our experiments that we conducted with the Aplysia model system about the role of Nox-derived ROS in regulation of growth con actin dynamics and neurite growth. And in the last part, I will focus on our studies that we conducted in the Zebrafish model system about Nox2 and how it regulates the development of these retinotectal connections. I would like to give credit upfront to the people that did the work. It started all with the work of Vidya Munamolai, a former graduate student in my lab that did these studies in Aplysia, which were then picked up by Corey Weaver, another graduate student in my lab, and he moved then these experiments into the Zebrafish model system, which were then followed by Haley Roeder, a former master student, and Adlan Terzi, a graduate student that just graduated from my lab recently. So what is axonal growth and guidance? So like myself, when I need to find my way to work, this is indicated by this path here on the aerial view of West Lafayette. The neuronal growth cone has to find its way to the target cell. So I need to read cues to find my way from point A, which is where I live, to point B is where I work. So that the growth cone needs to look for cues in the environment of the embryo. These cues can be different, it can be molecular, but also physical cues. Now, this work is relevant in a number of aspects. We would like to have a better understanding of basic normal nervous system development, indicated here by this diagram showing human brain development, but obviously also to better understand developmental disorders such as L1 syndrome, indicated here, or to develop better treatments for regeneration following injury and diseases such as Alzheimer's disease. Now, the neuronal growth cone is this highly motile structure at the tip of axons, as indicated here in the upper left panel. This is an image of a hippocampal neuron. in culture. This was recorded in the lab of my colleague, Dr. Peter Hollenbeck, and it shows the growth cone at the end of the axon. It's stained for F-actin in red and microtubules in green. Then on the right of that, we see a schematic of a typical growth cone with more details. We see the different domains, the peripheral domain or P domain, the transition zone or T zone, and then the central domain or C zone. These different domains have different distribution of the cytoskeleton. The F-actin cytoskeleton is particularly in the P domain in the form of phallopodia, indicated in the schematic, and lamellopodia. The microtubules are predominantly in the central domain, but they also penetrate into the periphery, as you can see in the schematic, and they interact with the F-actin cytoskeleton. And these interactions and both cytoskeletons are really important for neurite outgrowth, growth cone motility, and growth cone guidance. Then in the lower panel, we see three images. On the left, we see a low power phase contrast image of a cultured aplysia neuron. And then we see in the middle a differential interference contrast or DIC image of a growth cone and the different domains, again labeled with central domain, transition zone, and P domain. And we can see the nice structure of this growth cone. And on the right side, correspondingly, we see the cytoskeletal staining of this growth cone shown in the middle. So in red F-actin and in green microtubule staining. So how do aplysia look like? If you have never seen an aplysia, these are invertebrates, live in the ocean. They're really cool animals, and they've been used quite a bit in nervous system studies. Why? Because their nerve cells are very large, and people have used them to do electrophysiology. We're using them for imaging purposes because these growth cones are so large. You have seen on the scale bar that these growth cones can be as wide as 100 micrometers. This also allows us to do very nice quantitative studies, both with respect to dynamics, but also mechanics. Just an example of that is shown here. So this slide talks about measuring microtubule dynamics using the technique called fluorescent spectral microscopy, developed by Claire Waterman-Storer and Ted Salmon. And this technique is in principle very simple. It uses the low labeling of filament structures by incorporation of only a small amount of labeled subunits, such as tubulin in this case. And then you get a speckled appearance of the microtubule as indicated here in the lower panel. This is from our work. What you see here is a time-lapse montage where you see different time points of a single microtubule growing and shrinking and also translocating. So the time is going from left to right, and the distance is showing along the y-axis, and these time intervals are 5 seconds here. We color-coded the growing microtubule in green and the same microtubule when it shrinks in magenta. You can see the little dots, and an internal dot or speckle is providing a reference point for translocation. The addition of new dots provide us with the ability to quantify the rate of assembly or shrinkage. So I would like now to move on to our story about ROS signaling in nervous system development. So this diagram talks about ROS concentrations, their sources, and their effects. So there are both exogenous sources on the right and endogenous sources, and many are listed here, and both ultimately affect the concentration of ROS. So on the right, you will have high concentrations, on the left, lower concentrations, and these are also affected by antioxidant defense systems indicated here. So it's well known that at too high concentrations of ROS, you have impaired physiological function. You have oxidative stress, you have aging, you have diseases, and damage to the cells. Now, more recently, work from our lab and others have shown that if you have too low ROS, as shown here on the left, you also have impaired physiological functions. And some examples of that are decreased motility, decreased proliferation, defective host defense, and defective gravity perception. So it appears now that somewhere in the middle is the best for cells. So whereas homeostasis, where you have normal growth and metabolism and normal functioning signaling pathways. Now, what about ROS in the nervous system? Obviously, the brain needs a lot of oxygen, and therefore the brain is particularly vulnerable to oxidative stress. Secondly, it has been shown that NADPH oxidase-derived ROS have been implicated in neurodegenerative diseases such as Alzheimer's and Parkinson's and ALS, but also following injuries such as spinal cord injury. However, there's also evidence that ROS derived from NADPH oxidase play a role in normal neuronal functions such as differentiation, synaptic function, and plasticity, and gravity perception. So we were curious to ask the question, do ROS play a role in axonal growth and guidance? So moving on to the second part, now to our studies on ROS in growth motility and actin dynamics in the aplysia model system. Again, these were conducted by Vidya and Corey Weaver. So what are our hypotheses? So with respect to growth, we think the most simple hypothesis could be that intermediate ROS concentrations, as shown here in the middle in the green, are best for optimal neurite growth, while too high concentrations on the right and too low concentrations on the left will reduce neurite growth. With respect to growth guidance, we can envision multiple scenarios, but some simple hypotheses are depicted here. If you think about a repulsive cue indicated by the red gradient on the right side, the growth cone would turn away from that source. This could be mediated by an increase of ROS, as indicated here by the red color. On the left side, on the other hand, if you have an attractive positive cue, such a cue could introduce a small increase in ROS levels that actually has a positive effect. So depending on the level of growth cones in the cytosol of the growth cone, you may get either positive or negative effects. And that kind of correlates with the curve on the left side, the growth curve. This diagram talks about the molecular pathway that we envision for some of these responses. So it starts all with a guidance cue, for example, SLIT2, binding its receptor and starting an initial signaling cascade. This signaling cascade initially then activates the NADPH oxidase, in this case Nox2. Nox2 is a multi-subunit enzyme complex that has two components in the plasma membrane, in green here Nox2 and in purple P22, but they're not sufficient in this case because they're being activated by cytosolic subunits labeled here as P67, P47, and P40, as well as RAC in its GTP bound form. So when these 6 subunits come together, then the complex produces superoxide on the outside of the cell from oxygen. This quickly converts into hydrogen peroxide, either on its own or by enzymatic reactions. And then hydrogen peroxide can interact and react with the cell surface, but also go back into the cell through channels. Inside the cell, hydrogen peroxide can modify a number of proteins, possibly signaling molecules such as SARC, cytoskeletal proteins such as actin, which ultimately changes the dynamics and the organization of the actin cytoskeleton. So that's our hypothesis. Now let's look at some data in this part of the talk. So here I show you evidence that reducing ROS levels reduces F-actin. The top panel shows on the left again a growth cone schematic. We already have seen that. And then 2 images of a control growth cone under normal conditions. EIC in the middle and F-actin and microtubule stain in red and green respectively on the right side. On the lower panel, this growth cone has been treated with an antioxidant called PBN. So this is not an inhibitor, so we have to use millimolar concentrations to significantly reduce ROS levels in these growth cones. And the effect of that is seen here very clearly in the middle panel. You see the growth cone becomes flatter, phylopodia extend and ultimately stall and don't grow anymore. And also the central domain grows forward and the ruffles in the transition zones disappear. When you look under the hood at the cytoskeleton, you can see that the F-actin cytoskeleton is much diminished. You see that in panel B double prime. So there is less F-actin compared to control, and also microtubules extend towards the periphery. So major changes with the cytoskeletal mounts, rearrangements, and also the growth cone dynamics. When we then specifically inhibit NADPH oxidases with an inhibitor, in this case, the inhibitor is called VAS-2870, that's in the lower panel. We again reduce the F-actin content. So the top panel shows the vehicle control, the MSO. The IC on the left, F-actin stain on the right, typically done with colloidine. And then the lower panel shows another growth cone treated with 5 micromolar of this Nox inhibitor VAS-2870 for 70 minutes. So a significant reduction in F-actin content. And when we look at neurite growth, this is on the next slide, and here is quantified total neurite length per cell in 3 different conditions: medium control, DMSO vehicle control, and then the Nox inhibitor VAS-2870, in this case, 2 micromolar concentration treated for a day. So on day 0, before drug addition, the total neurite length per cell in these 3 groups are the same, very similar. But then 1 day in drug, you can see from the black bar that the neurite length is significantly reduced compared to 2 control conditions. Then we wash out the drug. In this case, the cells are treated for a full day with this dose. There is no reversible effect here, and the neurite stays short, while the control group still continue to grow. So Nox inhibition reduces neurite growth. So depending on the species, for example, in humans, we have 7 members of the family of Nox enzymes, and they're listed here. We have different numbers. They can be distinguished based on the activation mode. And so what you see on this schematic taken from a review by Bedar and Krause, you can see in the upper left the Nox1 complex, then in the upper middle the Nox2, on the upper right Nox3, and the lower level on the left Nox4, middle Nox5, and on the right Duox1 and 2. So all have these enzymatic subunits that does the electron transfer from NADPH onto a molecular oxygen, producing a superoxide, shown here in red. But they are different in the way they are regulated and activated. Nox1, 2, and 3 are regulated by cytosolic subunits. Nox4 is constitutively active, and Nox5 and the 2 duoxes are calcium regulated. Now, we were curious to see whether any of these are actually localized in aplasia growth cones. We were fortunate to find antibodies that are actually specific for some of these subunits. We got these antibodies from Mark Quinn, and 1 to Nox2, and 1 to the cytosolic subunit P40. Here are the Western blots showing that they're pretty specific to aplasia proteins. And then here in the middle and on the right, we see the corresponding immunolabelings. So these are slightly permealized cells. We see a punctile distribution showing that these proteins are both membrane associated, and as you can see in the case of Nox2, also quite present in the central domain on organelle membranes. Now, on the next slide, we have done a triple stain of these 2 subunits of Nox2 along with F-actin. So in the upper panel, you see each individual stain for the 3 different proteins, and then on the lower panel first, the triple color overlay, and then zoomed in that region of interest, first the dual overlay of Nox2 and P40, And you can see while labels are closed, there's not a whole lot of yellow here. There's not much co-localization. And this is likely because these growth cones have not been stimulated with any agent. But under the same conditions, same growth cone, when you look at P40 and F-actin, you can see that many labels for P40 are actually co-localized with F-actin. So that suggests that the cytosolic subunit P40 actually is associated significantly with the F-actin cytoskeleton under non-stimulated condition, which has also been shown for other cytosolic subunits in other cells. Now the next slide shows what happens when you stimulate the growth cone with a particle that's coated with a cell adhesion protein. We call this the restrained bead interaction assay to induce growth cone advance. In the upper left, you see a DIC image. You see that particle at the right, and it's held in place with a micropipette. You can see the dark shadow of that. And you can see how the central domain actually moves towards that bright particle. It's a glass bead coated with APChem. Then in the other images, you can see Nox2 P40 and the color overlay. And what you can clearly see here is that there's not only more of those two proteins around the adhesion site, but they're also co-localized significantly at this adhesion site, suggesting that the activation of Nox2 may play a role in this process here in adhesion-induced advance. And this is quantified here in the right panel for experiments. So the RBI area means the bead area, the adhesion area, where you have more co-localization than in adjacent areas. So moving on to the last part of the talk about the role of Nox2, now specifically in regulation of the development of retinotectal connections in the zebrafish. This is work by Corey Weaver, Helen Roeder, and Adrian Terzi. So why zebrafish? So Adhesion is great for cell culture studies, as I mentioned, because these cells are big. They're great for quantifications of time-lapse, fluorescence, and biophysical experiments. However, there are significant limitations in Adhesion as well. There is no knockout or transgenic lines. There's no in vivo imaging capability. They're difficult to breed, and there are invertebrates. On the other hand, zebrafish, they are genetically tractable. They are great for in vivo imaging. They develop fast, have many embryos. It's a well-established developmental model system, and they are vertebrates. So, just as an example of what you can do in zebrafish, this is an example of imaging in the living zebrafish brain of an embryo using a specific line that expresses fluorescent protein, GFP, in retinal ganglion cells, but only in a few cells, and this is called sparse labeling. The line description is given here on the right, and what you actually see here is you're looking at the axonal terminals in panel A, a couple, and then in panel B, in more detail at high resolution at different time points, 10-minute intervals, the growth cones and phyloproteodynamics of the retinal ganglion cell axons and growth cones in the optic tectum, indicated here on the left side in the schematic. That's where the retinal ganglion cells innervate the brain. So it's a very powerful model system and technique. We actually did this here with live sheet microscopy, which allows you to do high resolution imaging for extended time periods without harming the sample. Now, we started the studies by treating these zebrafish embryos with a Nox inhibitor, in this case, cholesterol, for a 4-hour time window indicated here in the upper left, 2.5 micromolar, then washing out the drug, and then fix and stain, or looking in a live fish in specific lines that express fluorescent proteins. And the results are shown here. First, the control without the drug. In the upper image, you see looking at the eyes, frontal view, the two eyes, and this is a line that expresses GFP in retinal ganglion cells under the Axonal 5 promoter. This is at 48 hours post-fertilization. So this is normal. You can see the optic chiasm labeled there. And the lower image shows 1 day later the innervation of the optic tectum by these axons. The detectors are labeled on both sides of the brain. Now, if these fish were treated with the Nox inhibitor celestrol, as indicated in the upper left, for 4 hours, then we see some major changes. The ganglion cell layer is widened, indicated by that bracket. You don't see the optic chiasm that well. It's still there. It's enhanced in the inset. And also the areas of the optic tectum that are innervated are smaller. And some of these effects can actually be rescued by treatment of hydrogen peroxide, such as the thickness of the optic nerve and the area of innervation in the tectum, but not the thickness of the ganglion cell layer. And this is quantified down here. On the left side is the width of the ganglion cell layers in control, and then celestrol is wider. It cannot be rescued by hydrogen peroxide. The middle graph shows the thickness of the optic nerve in control, reduced by celestrol, but rescued by hydrogen peroxide by adding back hydrogen peroxide. And the right panel shows the area of tectal innervation, reduced by Nox inhibition, but again partially rescued by adding back hydrogen peroxide. So drug experiments are great, but we also wanted to do more specific molecular manipulation by creating mutants using the CRISPR-Cas9 technique. So we created Nox2 homozygous mutant and then looked at the retinal ganglion cells, the eye, and again at the optic tectal innervation. So this is wild type. We see here the individual channels, the retinal ganglion cell label, CN8, nuclear stain, and the overlay. We targeted these guide RNAs to 2 sites. We call them guide RNA 1 and 2. 1 targets very close to the N-terminus, 2 a little bit further in, but both will create non-functional Nox. With both guide RNAs, when we look at homozygous mutants, we see significant changes in the retina, very similar to what we observed with the inhibitor. So the GCL is widened, and this is also quantified here at 2 time points, 48 and 72 hours. So with both mutations, with both approaches, we see a widening of the GCL. And then we look in the brain, in the optic tectum. Again, first control, so this is wild type. Without the mutation, we see the typical innervation in green here of the optic tectum. But then, with both guide RNAs, with both types of knocking out Nox2, we see severe perturbation of the innervation. So it's diffuse innervated and it's not compact to the normally developed optic tectum. And this is quantified by line scans here on the lower right for both guide RNAs. So the wild type would be the black line, and the 2 guide RNA, so the Nox2 knockouts, are shown by the red and green. So we also did some ROS imaging with genetically encoded biosynthesis, such as ROGFP2 or 1, indicated here. And first, we did some validation. So on the left side, lower panel, we are looking at the head of a zebrafish embryo. The 2 eyes are indicated. When you treat them with hydrogen peroxide, we get more signals. When we treat them with the reducing agent DTT, the signals go back, indicated here by warm colors. Then we also looked at cultured retinal ganglion cells and using the same biosensor. So on the left side, you see the IC images at time point 0 and treatment with the guidance QSLIT2 after 30 minutes, which actually causes a stalling of the growth cone. And SLIT2 treatment not only halts the growth cone, it also increases growth cone hydrogen peroxide levels, as indicated here in the graph in a double prime. So ROS levels are increased. However, in the mutant, Nox2 mutant, this doesn't occur. So SLIT2 increases hydrogen peroxide levels in a Nox2 dependent manner, and SLIT2 also halts the growth cone. Furthermore, if you do a guidance assay where you apply SLIT2 in a gradient fashion in an angle indicated here by the arrow, these are enlarged images of growth cones from cultured zebrafish retinal cells. So on the left, 10.0, on the right, 60 minutes later, you can see SLIT2 in the upper panel. You see the growth cone turns to the left, while in the lower panel, we add celestial Nox inhibitor together with the SLIT2 gradient, and then the repulsive effect of SLIT2 is abolished. So the growth cone kind of ignores SLIT, as you see on the lower right. And this is quantified again on the right. You can see that the normal effect of SLIT2, which is causing a negative angle, is abolished by the presence of Nox2 inhibitor. So the last set of data is shown here. This is in vivo data again, showing a retinal ganglion cell axon staining in the optic tectum, as well as DAPI stain in wild type, Nox2 heterozygote, and also a double heterozygote for Nox2, and the SLIT2 receptors ROBO2, which is called astride. So that's the double heterozygote. So when you quantify the area of innervation, you actually find that a Nox2 heterozygote fish, and that's shown here on the right, don't have significantly reduced area of innervation, as well as the astride heterozygote fish. And that's the green triangles. But in combination, the double heterozygote Nox2 astride has actually reduced innervation. This suggests that these two proteins, Nox2 and ROBO2, may be the same pathway. So this is the pathway we think about. SLIT2, the guidance Q binds to receptor ROBO2, starting a signaling cascade that ultimately activates Nox2, producing superoxide hydrogen peroxide, and then ultimately affects downstream target proteins such as cytoskeletal proteins and signaling molecules such as SARC, which ultimately affects axonal growth and guidance. So that's our current model. I would like to summarize and conclude. So first, I showed you that lowering growth cone RS levels by Nox inhibition reduces F-actin content, but also F-actin dynamics and Neurite growth. Then I showed you that pharmacological Nox inhibition in the zebrafish embryo inhibits the development of retinotectal connections, and this can be confirmed with Nox2 homozygous mutants. And then we also showed you some in vitro data with cultured neurons that Nox inhibition abolishes the guidance responses of RTC axons to SLIT2, but we also found that for other cues such as EDNF and NETRIN. So in summary, these findings suggest a role for Nox2-derived reactive oxygen species in axonal growth and guidance. Last but not least, I would like to thank the people involved here. So this is a recent picture of our lab. I would like to thank all my students and collaborators listed down here on the left side in this project. And of course, also all the funding sources from NIH, NSF, Indiana, Spinal Cord, the Injury Research Program, and also Purdue. And thank you very much for your attention. Thank you.